today we are here with Michael Schloter at the Helmholtz Center in Munich. Thanks very much that I could have come here to do a short interview with you.
Yeah, thanks, Andreas. We are very happy to have you here, and I think it's really very important to promote what we are doing in the MICROBES project. And we are also very happy, of course, to be part of this really great initiative to bring together molecular biology and also cultivation-based approaches of microbes, because I think this is really needed for the future. And therefore, we are very happy that Tanja set up this project and we got funds now from the European community. My name is Michael Schloter. I am here director of an institute at the Helmholtz Center called Comparative Microbiome Analysis. And since last year, I'm holding a chair at the Technical University in addition called Environmental Microbiology. And this already tells a little bit what we are doing. So we are interested in mainly environmental microbes and how they are influenced by climate change, by land use changes, by continuous urbanization, and all these threats that occur at the moment and which reduce biodiversity on a macro level. We have now also indications that the micro level, the microbes in the environment change. And we want to understand how these changes affect ecosystem services, but also how they have an influence on the human microbiome. Because what the human microbiome is doing, or what we as humans are doing, is we are recruiting a part of our microbiome from the environment. Meaning if there are changes in the environment, this is mirrored somehow also by changes in our microbiome. And changes in our microbiome can lead to a so-called dysbiotic stage, an unhealthy stage, meaning that many diseases might occur at this stage because they are linked to changes in the environment. And this is just cancer, it is metabolic diseases like diabetes, it's also allergies, it's infections. And we want to understand these interactions between the environmental microbiome and the human microbiome and how this influences our health.
Okay, thank you. You already answered a little bit also my second question, that what are you doing in the microbe project, but maybe you can further elaborate a little bit why microbe is such a cool project also for you and what you like to develop there.
So what we have been doing the last 25 years is we were all fascinated by these molecular approaches to describe microbes in the environment, which has not been possible before, because we all know that it is very hard to cultivate microbes. When you do a normal cultivation on a classical medium, you only get roughly 1% of the microbes that are present. So we were all fascinated by these molecular approaches. We generated tons of sequencing data on microbes, on microbial communities, on the dynamics of microbial communities, on their functionality. But we forgot somehow that we want to make use of this information. We need the microbes in hand. And this is exactly what microbes is doing. Microbes is based on this huge amount of data that is available. And we want to make use of this data to define new strategies for cultivation. And taking into account that in nature, for example, in 1 gram of soil, we have roughly 10,000 different species. We need approaches to find out which are the most important organisms. And there bioinformatics helps us a lot. And this is what we are responsible in microbes for, to define very important organisms for a particular ecosystem. We call them keystone taxa. And we want to use the molecular information that we have, not only to reconstruct the genomes of these organisms in silico, but we want to use this information to get these organisms cultivated. And for that, the interactions that we developed in microbes, meaning how to store samples that microbes can be cultivated. What does it mean if we have these organisms in hand? How can we implement them into synthetic communities? What are the legal issues behind that? Because once we work with the living organisms, there are things becoming more and more important, like Nagoya Protocol and other things, but also ethical things are becoming more and more important. So this is why I think microbes is such a fascinating project, because it's really interdisciplinary and it will really help us to push things a little bit forward, or a big step forward, to get organisms cultivated.
Okay.
And to apply them at further stages then for biotechnology, for human health, but also for sustainable agriculture.
Okay, thank you. I think that shows how important it is, this work, that it's really also successful. What do you expect for the next 3 to 5 years? Maybe here it's also to think about longer terms as results from the research, even after the MICROBE project.
Yeah, so we started here to make the interview here in this lab, and you might ask, what is this behind us? This is an artificial gut system. And what we have learned in the last years is that due to diet, due to lifestyle, the diversity of our gut microbiome was strongly reduced. And this caused a lot of problems, like invasion of pathogens, but also metabolic diseases, food allergies, and so on. So what we want to do is, we want to understand which organisms are missing in these particular systems. We want to get these organisms isolated. We want to bring these organisms back into the humans to substitute for what has been lost in the last decades. And then we want to see if this really promotes health. And this is what we are doing in the system. This is an artificial gut system where we take new microbes that we have been isolated, where we think that they are important. We integrate them into existing microbial communities, which we already have, so-called synthetic communities, and then we apply certain types of diet. And then we want to understand in the different gut compartments, how does this affect microbial communities, microbial functionalities, mainly also interaction pattern with the gut epithelium, because this is then the thing where the barrier to the humans is really taking place, and therefore we want to know if these new organisms, where we think that they are important, where we could show already in vitro that they are important, if they really behave in this expected manner also in an artificial gut system. And this is of course strongly required before we start doing experiments with animals, and maybe at a very later stage also with humans.
Okay, thanks very much. That sounds very interesting. Now maybe to close our interview, I think it would also be interesting, what are you doing when you are not in the lab or you're not doing research? Do you have other things to do in your life?
In fact, I'm really very happy that science is only one part of my life. It's a very important part of my life, but it's only one part, fortunately. At home I have my family, and I'm really much enjoying this family. We have kids around and they're all becoming great soccer players at the moment, so I'm trying to promote their soccer career as much as possible. Myself, I'm still doing, maybe not professional, but I'm doing skiing and ski racing. So there's a lot of different things, and I think this mixture of different things, this makes me also very happy in life, and I would not like to miss any of those parts. So I'm really very happy and enjoy life.
Yeah, that sounds very good. And yeah, thanks for the talk. It was very interesting here. And yeah, I think all the best for the MICROBE project also, and that you reach your goals also in life and research. Thank you very much.
Yeah, thanks Andreas for this really nice interview, and also for giving us the chance to present what we are doing, and we are looking forward for this interaction with you also in the future in microbes, but of course also beyond that.